Hello, Sugar Science. I'm Erica Kai from Indiana Bioscience Research Institute. I'm an assistant investigator at the IBI Diabetes Center, also at the Diabetes Center at Indiana University School of Medicine. So I established my lab in April back in 2021. Since then, my research has been focused to integrate genetic engineering with the beta cell biology, try to identify the new genetic target that we can use to help or assist this beta cell to survive better under stress or stress condition and also autoimmune conditions. So today I want to talk about one of our ongoing project, and this is focused on to using in vivo CRISPR screen to identify a transcription factor named ZBAT3 in the beta cell vulnerability. So I want to start my talk to using this very nice recent published review article in the Nature Reviews Endocrinology. So I want to pinpoint about from the current status about the clinical trial for the beta cell replacement therapies. So now we are able to use the induced pluripotent stem cell and derive the beta cell and the transplant into an individual living with the T1D. But this kind of first generation, the beta cell therapy required a combination with the systemic immunosuppressant. So this is right now it's at the clinical trial. I think the latest one has been moving to the phase 3, but the immunosuppressant, systemic immunosuppressant is still quite challenging for most of us. We can understand about what's the drawback from this. So as a T1D researcher, I think we are thinking about how to move into the next generation. So maybe we can use something to help the beta cell, such as encapsulation device, or is it possible we can generate some decellularized tissue structure that can help the beta cell to survive better under the stress condition. I think at the end, we are thinking out of all, it's the ultimate goal is to achieve the beta cell therapy to have a real cure that we don't need any suppressant, immunosuppressant for with to help the beta cell and also to make the individual they are able to maintain the physiological glucose level. So this is a slide just summarize the current challenging I have or I'm living in my mind and this here. I mean, this the broad immunosuppressant definitely is one of the biggest challenging, and also we are dealing with it right now. And also the side effect about the islet encapsulation. So this could be increased about the stress condition and also blocking see, you can imagine that in islet encapsulation, kind of you try to wrap the islet with a protected device. So islet is a tissue require a lot of the oxygen and the glucose exposure. So is there any way we can help them to able to have this still enjoy this kind of environment, but blocking out the autoreactive immune cell? So this is quite challenging, but also something I think in the field, the T1T researchers are very focused on this. The third point is about how we can improve the beta cell differentiation and also to increase the functional part of the beta cell out from the transcription differentiation process. It's any way we can try to reduce the cost about the stem cell therapy and also how this kind of duration can last about the insulin independent duration. At the end, also it's the safety concern about the strategy to using the immunoinvasive islets. So this is still the challenging, but I think back into the project, we want to talk about it, how we start, and also why we are thinking about to using the CRISPR screen for the beta cell immune tolerance or immune therapy. So this is need to be rewired time clock back into my postdoc time at Jocelyn. So my mentor Pony and also the Stefan Kissler, they actually it's kind of like a whole conversation. Yeah, thinking about is there's any way we can help the beta cell. At that time, we don't have real answer for this. So we are thinking, how about we employ a forward genetic approach, a screening approach. So we take advantage of one kind of cell line called one cell line, the beta cell line derived from NOD mice the type 1 diabetes mouse model. So using this cell line allow us to do autologous transplantation. We try to transduce the lentivirus like a CRISPR library to this like NOD derived beta cell line. So we prep this like a or generate this mutant cell line. They carry with mutation, loss of function mutation, targeting about 19,000 genes. We transplant this mutant cell back into the NOD skid, and with the control cell, without induced with autoimmunity, and also with our experimental group, we induced autoimmunity by injecting the diabetic splenocyte from NOD mice. After the 2 months, actually 8-week screening, without the selection pressure, we found we can retrieve the graft and found all the guide RNA which introduced into the mutant cells. They are still very well homogeneous presented. However, in our selection autoimmune group, we found all the 50,000 guide RNA, they are almost gone. Only 11 guide RNA they are able to survive. And 2 of them actually has been identified to associate with the human diabetes. So one of the names is called Rennes. So Rennes is associated with type 1 diabetes, human diabetes. And this story has been published back in 2020 in the Nature Metabolism. In the past 2 years, we also have follow-up study and published in the diabetes and one in the bioarchive right now. So today's project, we want to focus on the second, the human T, the diabetes gene. It's called ZBET3. So what is ZBET3? ZBET3 is a zinc finger bed domain a domain containing protein. So this protein, along with its family, has been projected as a transcription factor due to they have this DNA binding that domain. But the exactly function for the ZBET3 is still very largely unknown. In the past, through the multiple GWAS publication, ZBET3 has been associated with the T2D substitutability. So why a T2D GWAS gene will show up in our T1D screen? So to validate this result, we first generate the ZBET3 mutant cell using the same guide RNA that is very highly enriched in our screen. And we also generate the control cell using the non-targeting guide RNA to do our experiment. We transplant the mutant cell and also this control cell back into the NOD skid using the same screening model and try to see if this is the ZBET3 mutation really can help the beta cell to survive under the autoimmune condition. In fact, yes, we found that. In the autoimmune group, when we try to induce, you see the ZBET3's mutant cells still are able to survive, while our control cell, they are rapidly eliminated by the autoimmunity we induce through the diabetic NOD splenocyte. We also move to the next to see if the primary islets can also repeat the same thing. We try to introduce the ZBET3 mutation by the guide RNA driven by the insulin promoter, and we transplant the islet onto the under the kidney capsule and do the same diabetes like an autoimmune model by injected diabetes NOD splenocyte. We found that the ZBET3 mutant islet they show still have more resistant to this autoimmunity by showing they have the enhanced insulin the RNA level compared to our control. So the next, we want to see if this kind of result can be translated into the human cell. This is part of work we collaborate with Dr. Milton Lab at Harvard Stem Cell Institute. So we're using the T1D donor iPSC cell, generate control, and also the ZBET3 knockout cells to stress this 2 cell line to control and the ZBET3 using the ER stressor, sepsic organ. We found a control cell, they are very sensitive to the stressor, show increased apoptosis level after 24 hours treatment, while our ZBET3 cells show very diminished response, and they show the significant improve their survival under the stressor. In the transcriptional data, we also found the same thing. The ZBET3 they show they are more increase their survival by suppressing caspase-3 apoptosis pathway. They also show less response to the UTR, the UPR, the unfolded protein response markers. But most of the things that's very quite interesting is in the wild type or the control, the human stem cell derived beta cell. We found the cell, they are highly sensitive to the stressor by showing upregulation about the surface MSC class 1 molecule, HLA ABC, while our ZB3 show very diminished kind of response to that. This actually can be in line to the mouse, the beta cell line RNA sequencing. So in our mouse mutant NIC1 cell, when we compare to our control under the stress condition, our top one downregulated gene, actually the pathway is called class 1 MSC mediated antigen processing and presentation. So this makes us wonder what is exactly the functional role of ZB3. So to investigate the role of ZB3, we have to conduct a chip sequencing, the chromatin immunoprecipitation sequencing. This is collaborating work with Travis Johnson, who is director at the IBRI Data Science Center and also a professor at the IU School of Medicine. So we identify the significant ZB3 binding site, but most interesting is the most top hit mutated target of ZB3. They are all involved in the interferon pathway. This all the pathway target, mutated target, when we back into look into RNA sequencing, we do found there are very little response to the stressor, while the control, you can see all the mutated target, they are quite response to the stress treatment. To validate this result, we also generate a new mutation line to disrupting the ZB3 expression focus on the DNA binding domain. We're using the dual guide RNA strategy to target on the BAT binding domain, and then we validate this by the Western blot and also the functional assay with interaction with the diabetic NOD CD8 cells. So first of all, we found state 1, state 2, IF1. This is top 3 mutated target. We identified from our chip sequencing. We found they all show very little expression level in our ZB3 knockout and also the bad domain deletion, the mutant cells compared to our control. The cells, they also show reduced the surface MSC class 1 expression. When we culture this with autoreactive T cell, purifying from the diabetic NOD cells, we found the T cell, they show very diminished and very little response to the beta cell by standing by the CD25, the activation marker. At the same time, we also try to see if we can recapitulate this kind of effect in the human beta cells. We are using a differentiated stem cell derived beta cell and to transient silencing the ZB3 using the antisense oligos ASO treatment. After 2 days of transient knockdown, the ZB3, we found the surface level of the HLA class 1 level. They show significantly reduced induction compared to the control upon interferon gamma treatment. And now we are working with the Roberto Malone lab and also 7U lab to see if this kind of ZB3's knockdown or knockout effect can help them to be survive or improve their survival under the HLA restricted the CD8 killing assay. So hopefully I try to convince you using today's the presentation. We lost the ZB3 in the beta cell. They show this kind of beta cell show more resistant to autoimmune killing in the mouse model. And for both human and mouse cell, they show reduce the surface MSC class 1 level expression. And this way they are more restricted to suppress the CD8 T cell activation. The cell, they also show they are resistant to stress and also cytokine treatment. I don't have time to talk about the function part, but in fact, the ZB3 mutant cell, they show enhanced insulin gene expression and also secretion. So this might be a part of they also involved in the phosphomediation, the transcription. Is also another pathway we identify from our ChIP-seq. So I want to use the final using this slide to highlight how the beta cell or the cell immunogenicity and class 1 expression got regulated by the signaling pathway. So from the ZBAC3, we identify its putative target, including the IF1, state 1, state 2. State 1, state 2 work together, or the state 1 also can form the dimer to activate the gene that evolved to, at the end, to express the MS class 1 gene. These three transcription factor or mediator downstream of the interferon pathway, they also are able to reciprocally activate each other. So this is form a very complicated a pathway or regulation to each other. And then our ZB3 actually, from our chip sequencing, identified the target of the three and also involving other genes. So at the end, what I actually want to highlight is the complexity of transcription and regulation of beta cell immunogenicity. I think using this two and also why the ZB3 one transcription factor are able to activate so many mediator that involved in the antigen presentation, MS class 1 active expression. I think this might be tell us a story about how the beta cell response to stress and this kind of complexity might be through the evolution. It's kind of important to reserve and preserve things you see the cell. They are trying so hard to have all the mechanism or all the factor play around and along together, try to make sure how the immunogenicity or this mechanistic part is intact. So I hope I can try to use the story to talk about all the collaborative work between the IBI Diabetes Center and also the IU, the CDMD Center for Diabetes and Metabolic Disease. We are the neighbors. We are very good neighbors. And then through the collaboration, I think we all want to increase or enhance our translational potential for the diabetes research. So at the end, I want to thank you for my lab and my lab people, and particularly for Jialing Wu, who is the one contribute the most to this ZB3 project, and all our collaboration through the IBI, and also the IU School of Medicine, and also my previous mentor and collaborator. And also want to thank you my funding through the Breakthrough T1D. So thank you so much.